Welcome, curious minds, to another exploration designed just for you. Today, we're tackling a question that's really vital for so many people navigating mental health challenges. Why does ketamine seem to work wonders for some with severe depression, but leave others still struggling? And what if the answer lies in something as fundamental as, well, inflammation?
It's a truly pivotal question. And the research we're unpacking today offers a remarkably fresh perspective, urging us to look beyond just, simple labels of responder and non-responder. It helps us understand the deeper biological mechanisms really at play.
We're drawing our insights today from a pretty groundbreaking paper by Norihiro Okada and colleagues. It's titled Intron Retention, a novel method for evaluating the response to ketamine in patients with treatment-resistant depression, published in 2025.
Yeah, this study is really pioneering because it uses a completely new lens to examine what truly differentiates responses to ketamine. It delves into the fascinating and often overlooked world of our body's cellular responses to stress. It offers clues at a molecular level we haven't really seen before.
So get ready as we unwrap this cutting-edge research. We want to offer you a shortcut to being exceptionally well-informed about ketamine, the complexities of depression, and, maybe surprisingly, a connection to how our bodies manage stress and fight infection. Let's begin by grounding ourselves in the challenge: major depressive disorder, or MDD. It's a debilitating condition, casting a long shadow over both mental and physical well-being for millions. But for a significant portion, maybe about a third of patients, traditional treatments, antidepressants, psychotherapy, they just don't provide enough relief. We call this particularly stubborn form treatment-resistant depression, or TRD.
And that's precisely why this research is so critical. As we've touched on in past discussions like ketamine's real-world effectiveness and ketamine and neuroplasticity, TRD isn't just a label. It's an incredibly challenging reality. It brings high medical costs, a severely reduced quality of life, and unfortunately, a heightened risk of relapse and even increased mortality. The devastating risk of suicide being a most serious concern there. And despite, decades of research, our understanding of why some people develop TRD and how to effectively treat it is still, frustratingly incomplete.
Right, and this is where today's study introduces this really fascinating, maybe slightly technical-sounding concept: intron retention, or IR. It sounds complex, but it's actually a natural process in our bodies. Can you break down what IR is and why it's such a potentially powerful new tool here?
Certainly. Think of your genes like instruction manuals for building proteins your body needs. These manuals have vital sections, exons, and then these sort of filler sections, introns, that usually get snipped out before the final protein blueprint is made. Now imagine your body's under stress, maybe fighting an infection or dealing with significant psychological pressure. In response, your cells might decide to keep some of those intron sections, at least temporarily. That's intron retention. It's a molecular mechanism that helps cells adapt, maintain protein balance, kind of fine-tuning the instructions to cope.
So it's not a mistake. The cell is doing this deliberately in response to something going on internally.
Exactly. It's a deliberate adaptive response. And the brilliant part is, by identifying which specific genes undergo this IR process and how much, we can get really nuanced clues about the type and severity of the stress the body is experiencing. It's almost like a sophisticated cellular stress barometer.
A cellular stress barometer. I like that. So it tells us not just that something's wrong, but maybe what kind of internal battle is going on. And this study used that principle to look at existing data from TRD patients, analyzing these IR genes to get a whole new perspective on how they responded to ketamine, like rereading a book with a new magnifying glass.
Exactly right. That's a great way to put it. And that approach is key. They didn't start from scratch, recruit new patients. Instead, they did some brilliant scientific detective work. They reanalyzed existing RNA sequencing data from a previous, well-regarded study by Ketamine and colleagues. This let them unlock new insights from data that was already out there.
Okay, so tell us about that data. Who were the patients involved?
Sure. They looked at data from 21 healthy controls and 26 patients diagnosed with TRD. Now, among those TRD patients, 18 were classified as responders to ketamine, meaning their depression symptoms got significantly better, and 8 were non-responders. And importantly, they had data from these patients both before and after getting ketamine.
Okay, but only 8 non-responders. That sounds like a small group.
It is, and it's worth noting the authors are very upfront about this, that small sample size, especially for non-responders, is a limitation. It's common in these kinds of early groundbreaking studies. It means the findings are compelling, definitely, but they'll need checking in larger groups later on.
Right. And what did they do with this raw genetic data? You mentioned RNA sequencing. It sounds like they had to clean it up quite a bit first.
Oh, absolutely. Think of it like sifting through tons of sand to find tiny diamonds. They meticulously process the raw RNA data, which is
Yeah.
basically a snapshot of gene activity. This involved several crucial steps: removing low-quality segments, filtering out any non-human genetic material, contamination, and then aligning the good data to the human reference genome.
Hmm.
This thorough filtering isn't just like housekeeping. It's essential to make sure the signals they detected, especially for intron retention, were real and biologically meaningful, not just noise.
Got it. Foundational work. So, with this powerful new IR lens applied to that clean data, what differences started to pop out when they looked at the non-responders before any ketamine was even given? What was already going on?
This is where it gets incredibly insightful. Even using traditional gene expression analysis, before getting deep into IR, they found non-responders showed signs of elevated innate immunity. Now, your innate immune system, that's your body's first-line defense, right? Always on alert.
Mm-hmm.
In non-responders, this system was particularly activated, especially genes related to viral infection. And this actually lines up with previous reports suggesting activation of something called the type I interferon signaling pathway in MDD patients. Think of that pathway as the cell's alarm system for viruses.
So even before treatment, non-responders seemed to be fighting some kind of internal inflammatory battle, specifically one that looked like a viral infection response. How did the new IR analysis add to that picture?
The IR analysis was even more striking and more specific. It didn't just confirm the general inflammation. It showed significantly more changes in intron retention in non-responders compared to responders. And again, overwhelmingly, in genes related to innate immunity and viral defense. It strongly suggests non-responders have significantly elevated and active innate immune responses, very likely due to an ongoing viral infection. They specifically pointed out IRF5 and CXCL10. IRF5 is like a master switch for antiviral responses. CXCL10. Think of it as a potent chemical siren calling immune cells to a viral attack site.
Wow, so these signals were really strong in non-responders, painting a picture of a body already under siege, maybe even without obvious symptoms of being sick.
Exactly, a significant underlying biological state.
So non-responders are dealing with this intense inflammatory state. What happens when ketamine actually comes into the picture? Does it do anything for them, even if their depression doesn't lift?
Right. This is where the study delivers a really remarkable twist, something that challenges older ideas. They found that ketamine administration actually restored the expression of several key inflammation-related genes. It brought them back towards healthy levels. And here's a key bit. It did this in both non-responders and responders.
Wait, really? In both? So, regardless of whether someone felt better depression-wise, ketamine was having a biological effect on their inflammation.
That's what the data shows.
That's fascinating. So, ketamine is having an anti-inflammatory effect even in the so-called non-responders. Can you give us some examples of these genes? What are they doing?
Yes, absolutely. For non-responders, genes like CXCL10, that siren we mentioned, and MCM, which helps immune cells move around, showed restoration. Their activity was brought closer to normal. Also, IDO1, another one restored in non-responders, is a key immune regulator, often hyperactive in chronic inflammation. In responders, other genes like MUC1, MUC5B, and MUC6 showed similar restoration. These MUC genes make mucins think protective slime layer on cells, forming a barrier against viruses and regulating inflammation. So yeah, many of these are key players in protecting against viruses by modulating cytokine expression, the signals immune cells use to communicate.
Okay, so even if the depression symptoms aren't lifting, ketamine is actively helping calm this underlying inflammatory storm, trying to turn down that cellular alarm system, regardless of the clinical outcome for depression.
That's precisely what the data suggests. It implies ketamine restores this cellular inflammatory response, at least to some extent, in everyone. And actually, what's even more striking, the study's analysis showed the effect of ketamine on the recovery of inflammation was potentially even greater in non-responders, based on the network of protein interactions they looked at.
This really challenges our whole idea of a non-responder, doesn't it? If ketamine is doing positive things for inflammation in these people, why aren't their depression symptoms improving? What's the study's big idea here?
This is the study's core groundbreaking hypothesis. They propose that TRD non-responders don't have a constitutional resistance to ketamine. It's not that their bodies just ignore the drug. Instead, their pre-existing, extremely elevated inflammatory state, that intense, likely virally driven immune activation, it just overwhelms ketamine's beneficial effects on mood. Essentially, ketamine is working biologically, but the inflammation challenge is so huge that its antidepressant impact gets kind of drowned out. Ketamine, in these specific cases, is insufficient, not truly ineffective.
Insufficient, not ineffective. That's a really profound shift in thinking. It completely reframes the narrative around treatment resistance. It implies, well, if we could tackle that underlying inflammation first, maybe with other therapies, ketamine might then actually work for their depression.
Precisely. It moves us away from blaming the drug or the patient's biology in a simple way and towards understanding the complex biological context the drug is working in. This really is a pioneering exploration into molecular details differentiating responders from non-responders. It's much more nuanced than just yes or no.
Okay, so if this hypothesis holds up, what are the potential real-world implications? How might this change how we approach TRD and ketamine therapy down the line?
Well, it points towards a future of much more personalized medicine and mental health. Instead of just labeling someone a non-responder and moving on, we might identify them as having this specific high inflammation profile. And that might need additional targeted support. It could lead to innovative combination therapies. You could imagine using specific anti-inflammatory treatments, maybe even antiviral drugs, alongside ketamine.
Right, the idea being to first lower that overwhelming inflammatory burden.
Exactly. To clear the way, allowing ketamine to then exert its full antidepressant potential without being swamped.
It sounds incredibly promising, genuinely offering new hope. But I imagine a study like this, especially with a novel method like IR, it must have its limitations too.
Oh, absolutely. And the authors are commendably transparent about this. As we mentioned, the sample size is relatively small, 27 patients total, only 8 non-responders. That makes it tricky to reliably adjust for other factors like age or sex, which can influence both inflammation and depression.
Sure.
However, it's important to stress the IR analysis method itself is considered quite robust. And they did perform rigorous sensitivity checks that confirmed their main conclusions. That adds strength despite the sample size. But absolutely, further validation in much larger, more diverse groups is essential to really confirm and generalize these exciting results.
Okay, so this has been incredibly insightful. We've learned that for people with treatment-resistant depression, a key factor in whether ketamine appears to work might not be resistance to the drug itself, but rather an underlying extremely elevated inflammatory state, possibly linked to something like a persistent viral infection.
And critically, ketamine seems to partially restore this inflammatory balance even in those non-responders. It redefines non-response not as a failure of the drug, but maybe as an issue of its effects being insufficient to overcome that massive biological hurdle.
Not ineffective. That really opens up possibilities for the future of mental health treatment. It suggests a more targeted approach, one that addresses individual inflammatory profiles, could unlock ketamine's potential for many more people.
It really pushes us to think beyond just single drug therapies, doesn't it? To consider combining treatments in smarter ways, personalizing care to overcome specific biological hurdles that until now were basically invisible.
Okay, here's a final thought for you, our listener, to consider. If understanding individual inflammatory profiles, maybe identified through methods like Intron Retention, can revolutionize how we treat depression with ketamine, what might this signal about the future of mental health care overall? Could routinely assessing these unique biological markers lead to truly personalized treatment plans that move far beyond a one-size-fits-all approach? Not just for depression, but perhaps for many other chronic conditions that have long baffled us. Thank you for joining us on this exploration. Until next time, stay curious.